Ladies and gentlemen, I'm Dr. Juan from Shanghai Changzheng Hospital, China. On behalf of co-author, I'm happy to present our data. Updated results of a multicenter first-in-human study of BCMA and CD19 dual-targeting FasT CAR-T GC012F for patients with relapsed and refractory multiple myeloma. GC012F is a second-generation BCMA and CD19 dual-targeting CAR-T, which was designed to overcome antigen escape and to travel faster, deep, and durable response in myeloma patients. As we know, BCMA is a universally expressed on malignancy plasma cell and a well-established target for multiple myeloma. CD19 is expressed on both myeloma cells and their progenitor cells. It has been reported from previous study targeting CD19 can trigger elimination of malignant cell of CAR-T. This CAR structure of this drug product enables stable CAR expression and effective functionality. GC012F is manufactured on the novel FasT CAR platform, enabling next-day manufacturing. The platform was designed to overcome major challenges of conventional CAR-T therapy, one of which is the lengthy wait time between apheresis and the infusion for patient with progression. Another potential advantage of FaSTCAR manufacturing is enhanced T cell quality with less exhausted T cells. This is our study design. This is multicenter, open-label, single-arm IIT study. We treated 29 patients into 3 times 10 to the 5th power to 3 times 10 to the 5th power per kilogram. We are reporting data with the day cutoff June 8th of this year. Here is our eligibility date criteria, which is similar to others. The primary endpoint was safety, and the second endpoint include ORR, best of response, MRD assessment, PK, and PD. This is the patient's characteristics. Notably, 90% of patients evaluated of a high-risk profile according to mSMART 3.0 criteria, which 10% double hit and 28% extramedullary plasmacytomas. The median per line of therapy was 5. They are very heavily pretreated patients, with 7% of patients were triple exposed, with 93% PI refractory, 93% IMiD refractory, and 34% patients received dial-based therapy. 83% of patients were refractory to their last therapy. This is the ORR assessment. We observed 93.1% with the best response achieved over PR. 76% patients achieved CR, SCR, and patients achieved VGPR or better. Median duration of follow-up 11 months, ranging from 4.9 months to 34.5 months. Important to note, during the past 5 months of the follow-up, due to the COVID-19 situation, not all patients could attend their scheduled follow-up to visit reassessment deep of response. Therefore, the best of overall response in some patients might have gotten deeper. Median duration of response at the time of the cutoff was 50.7 months. Here is the MRD. 100% patients achieved MRD negative either by EuroFlow or by flow cytometer. There was no difference observed in dose level. This is a safety profile, which is similar to previous finding. It shows mostly low-grade CRS, which is 40% no CRS. 48% of patients occurred grade 1, 31 patients occurred grade 2, and only 2 patients occurred grade 3 CRS. No grade 4 or 5 CRS and no ICANS or neurotoxicity were observed in our patient. Most common hematological adverse events were neutropenia, lymphopenia, leukopenia, and thrombocytopenia. In terms of pharmacokinetics, GC012F FasT CAR-T expanded very well in all patients with long persistence in all dose levels. CAR-T median Tmax was day 10, and the median peak copy number was 97 copies per microgram genomic DNA. In conclusion, GC012F continues to show a favorable safety profile with mostly low-grade CRS, just grade 0 to 2. Notably, highly overall response rate, ORR can get achieved 93.1%. It deserves to point, MRD negativity achieved in all retreated patients. It can achieve at threshold 10-6, the patients get 100% MRD negativity. GC012F is a faster, deep, and durable response with a median DOR of 50.7 months. And still some patients still in follow-up. Together, GC012F is a dual targeting BCMA CD19, shows a very promising activity in relapsed and refractory multiple myeloma, including high risk and heavily pre. And finally, we would like to thank the patients and their families, all participating study centers and Grazing Biology for providing drug product. Thanks all the teamwork and thank you very much.